examples of really taxonomically broad phenotypic convergence in a particular habitat, we typically take that as pretty compelling evidence that there is some fundamental adaptive value of that convergent phenotype for that habitat. So the classic example of that would be white sands. We've got all these crazy critters at white sands walking around with the blanched dorsal coloration that now all of the now classic work in white sands lizards have demonstrated a really important role for selection in maintaining that phenotype, particularly in the face of ongoing gene flow from neighboring darker substrate populations. Perhaps less well known than white sands leucism is kind of the yin to that yang, which is salt marsh melanism. And this is an equivalently taxonomically broad assemblage of organisms living in brackish or salty tidal marshes, all of which have evolved enhanced melanism relative to their closest inland sister species. And a lot of them are birds, many of them are mammals, moles, voles, shrews, herps, snakes, and the melanic phenotype takes on a couple of forms, some of which are exhibited in combination by each organism, but it's a blotchy, like an enlargement of any melanic patch or skin or feather patch, or it's an overall duskiness, sort of a whole body melanic phenotype, or a combination of both. And because of the way that we assume convergence is pointing us towards some fundamental adaptive value type situation, this spawns a lot of hypotheses about, well, maybe is this also an example that suggests salt marsh organisms are under selection for background matching? And there is some evidence that tidal marsh sediments are darker than freshwater sediments. That's true for a subset of tidal marshes, but not all. And in terms of birds, there's been a lot of adaptive hypotheses for salt marsh melanism that revolve around the enlargement of plumage badges that signal dominance. That's helpful for coastal birds. And duskier gray, more melanic feathers confer a protective benefit in human habitats that have a lot of bacterial degradation of feathers. However, as you can see, those only work for birds, and the sediment is not broadly explanatory across all of the tidal marshes that these melanic forms inhabit. And so today I'm going to talk to you about some work that I've been doing in this species, the swamp sparrow, which does exhibit the melanic phenotype. And through some other work that we were doing, we had an alternative hypothesis pop up that may potentially have that broad explanatory power across the taxonomically broad assemblage. So in terms of looking at local adaptation in swamp sparrows, what I have been doing is comparing inland to coastal forms. They differ in a suite of phenotypes. Today I'll just talk about melanism and tolerance for salinity, but they are phenotypically different. Coastal bird is darker, and it's able to breed in these saline habitats. The tidal marshes, particularly in the Delaware and the Chesapeake Bay, but elsewhere in North America particularly, they really are extreme habitats. Almost anything a bird could eat or drink in that habitat has a higher salt content than a typical avian kidney is able to process. And so they have to have specialized physiological adaptations to living here. And that's true not just for these birds, but for some of the other species I showed in the previous slide. And the other reason to sort of start looking at some of these coastal adaptation questions in swamp sparrows is that they are extremely robust as an example of true adaptive divergence. We know a lot of things about them thanks to decades of work on the system, mostly by some of my co-authors in the work. Not only are they phenotypically distinct, the traits do confer a fitness benefit. That's been robustly demonstrated in the field and in the lab. The traits have a genetic basis, and that includes this melanism trait. There have been common garden experiments demonstrating that. And that selection actually does drive evolutionary divergence between inland and coastal forms. There are a couple avenues by which it does that, which I'm not able to talk about today. But they are truly an adaptive divergence case study, and they're very tractable for genomic applications to that type of question. They're very recently diverged, so these coastal habitats did exist at the last glacial maximum, so they're post-glacial. There's a lot of gene flow and hybridization at the contact zone between coastal and inland forms in northern New Jersey. There's very strong selection, so what that sums to is that signatures of selection are highly detectable in the genome against a very low homogenized genetic system. So what I did was to compare 30 inland and coastal birds and assemble a new reference genome based on a coastal swamp sparrow, resequencing those individuals to at least 5x depth and mapping them back to this reference, applying really stringent QC, allowing no missing data. And this is what the distribution of divergence across the genome of inland and coastal swamp sparrows looks like. The first takeaway is that these things are super similar. Genome-wide FST as one metric of differentiation is 0.015. But there are, as you can see, these very defined and detectable peaks across the genome corresponding to some regions of divergence. And so when we zoom in on these peaks, they're not spurious FST peaks driven by some other processes that we know can actually drive FST higher in a heterogeneous pattern. There are very robust hallmarks of a recent selective sweep of the majority of those peaks across the genome. They correspond, in terms of these two traits I'm going to talk to you today, they very directly implicate at least 28 genes involved in salt tolerance and melanogenesis. So these hallmarks that I'm showing on the left in this figure, genomic position across the bottom, and then divergence in some suite of metrics in relation to this known salt tolerance gene, for example. You have the FST track in black, and to note it's really high resolution because of that high level of gene flow. The peak itself really specifically does implicate an intronic region or intergenic region. It allows us to have a lot of confidence in which we parse through known functional genes nearby to divergent regions. Other hallmarks of a recent sweep include a recent purge of nucleotide diversity in the region. Tajima is D tanks, indicating a huge number or an overabundance of rare alleles following a sweep, and then it also coincides with runs of homozygosity. And so all of the things in this list have signatures like this in the genome, and they are largely genes involved in salt tolerance that fall into different physiological categories, the largest of which is vesicle trafficking genes. And then there are also genes under these peaks that are exclusively implicated for melanogenesis pathways. But something very interesting was that several of them have pleiotropic effects on both pathways. And so I'm going to highlight just one of those for you. That's this block1S2 gene. It's a subunit of the block1 complex, which is critical in mediating vesicle life cycles in the cell. And vesicle life cycle pathways affect many different endpoints of cellular function, and that includes vesicles that become lysosomes or vacuoles that sequester things like salt and aid in salt tolerance and excretion rates in the kidney. But this same complex, block1 in blue, mediates that process and the maturation and migration of melanosomes to pigmentation targets in the skin. So this one subunit has critical roles in both salt tolerance and melanogenesis. So in mice, when they tinker with block1, if you knock it out, you don't get normal kidney excretory function, you also get a change in the phenotype of the animal. So this, of course, raises the question of pleiotropy and its effect on this shared salt tolerance and melanogenesis phenotype we observe in the assemblage. And the question to me became, is this an example of adaptive pleiotropy, where melanism we see across these organisms is itself adaptive, but the pleiotropy with the intrinsic pleiotropy of vesicle trafficking simplifies and strengthens selection on both traits? Or could this be a molecular spandrel where melanism itself is not adaptive and it's a mutual consequence of tinkering with salt tolerance ability via this vesicle trafficking gene? So I chose to test this in swamp sparrows using an association study. We've got this very active zone of admixture, intergenerational hybrids. Everything's jumbled up in the genome in a good way for GWAS. So we took, so I think I added about 60 birds to that from the hybrid zone. And then we scored the melanic phenotype as a composite of traits. So not just overall dark, but looking at it in composite, there's the extent of black feathers. This is looking like top down on the bird's head. How far does the black patch extend along the crown? How dark is the skin of the bird? We scored off the legs, tail color, bill color. And there's some variation in each of those traits throughout the hybrid zone. And we excluded everything that wasn't biallelic, and we're sampling about one SNP every 260 base pairs. So running that either in Planck or GEMMA or both ways, we get the same results. I'll start with this crown phenotype. This gene here, BNC2, there's a strong association between the allele you have at this SNP adjacent to the gene and how much black you have on the crown. If you are homozygous for this A allele, you have more than 40%, and if you have one or both copies of the G allele, you have less. And this doesn't really vary very quantitatively when you score it across a larger number of individuals. You see it's a little bit binary in that way. So this is an example of a fairly strong, so the significance threshold that I'm setting for this comparison is via the Wald test, and this is a 10 to the -5, appropriate for the number of markers that we have in the study. And BNC2 is a zinc finger transcription factor that acts specifically within melanocytes. It's also one of the primary determinants of skin color in humans. We have a significant association between the salinity of the breeding habitat and the allelic state at these sets of SNPs right near the block 1 complex. So an association between the block 1 state and your ability to breed in a salty habitat or not. And what's extra interesting is you also have a significant association at a slightly more distal site to block 1S2 that is associated with the color of your skin as scored off the legs. So if you have the A allele, you have really dark legs. Oops, I'm sorry. And if you have the G allele, in terms of the one bird we saw with that phenotype, or with that genotype, you have really light legs, and the heterozygotes are intermediate. So the summary in terms of swamp sparrows, what we see from those data are that there is evidence that melanism in a swamp sparrow can be the product of positive selection. BNC2 seems to be explicitly implicated in the size of that plumage badge, which is under divergent selection, inland versus coastal, for reasons of intrasexual and intersexual selection. There's a trade-off between the orange part of the crown and the black. So this is an example of positive selection on a melanic trait, but we also have an example that suggests melanism in swamp sparrows can be a byproduct of selection for salt tolerance. SNPs in the block 1S2 region do associate with both salinity and melanism, but it's a different subset of SNPs, so it's perhaps not pleiotropy in the strictest sense, or that one that I might have expected. So two alternative lines that we're currently exploring are whether or not those two sites could have swept together on a haplotype, and that would explain selection on that for salt tolerance, would explain how abundant it is in the present day, or that our GWAS is not able to detect both with equal strength. And finally, in terms of the broader context, that vesicle trafficking genes are inherently pleiotropic, molecular pleiotropy becomes a parsimonious alternative to an adaptive hypothesis about convergence across the assembly. And even if it's not a great example, or if it doesn't prove to be a great example of convergent adaptive evolution, perhaps it's a really interesting case study in convergence via pleiotropy. And thanks to all the units that made that happen. And I think it's time for the next speaker.